Welcome back, friends. This is Dr. Mlelwa Lecturers. Don't forget to click the subscribe button and to click the bell, then put all for more updates. To join our Telegram group for discussions, just click show more in the description. Then if you scroll down, you see the links for the O-level and the A-level group. Welcome back, students. This is Advanced Biology Coordination, and we have the session 28. The facilitator in this session, I am Dr. Mlelwa, and in the previous session we discussed with the excitatory synapse, spatial and temporal summation at the rules of the synapse, and today in this session we are continuing with the inhibitory synapses and the neuromuscular junction. So if you understood about the concept of excitatory synapse, I said in the previous session kwamba excitatory synapse ni zile synapse ambazo the binding of the neurotransmitter at the postsynaptic membrane inapelekea inside of the cell or inside of the neuron to become positively charged. While I explained that the inhibitory synapses the binding of neurotransmitter at the postsynaptic membrane inasababisha the inside of the cell to become more negative with respect to the outside. That means cell hiyo haiendi kutengeneza action potential. So actually synapse inakuwa imeinhibit the generation of the action potential. So hapo sio neurotransmitter, that is synapse. So the same neurotransmitter such as acetylcholine inaweza ikawa ni excitatory in some of the synapses, and it can be inhibitory in other synapses. So inhibitory synapses, at inhibitory synapses, the release of transmitter substance increases the permeability of the postsynaptic membrane by opening up ions specific channel to chloride and potassium ions instead of sodium and potassium ions. So as we know that the concentration of chloride ions outside the axon is higher as compared to the inside, so the opening up of the chloride ions will cause the rushing in of the chloride ions, and the chloride ions zitakapokuwa zinaingia zitasababisha the inside of the cell to become more negative. So as the chloride ions rush and potassium ions rush out, chloride ions rush in and potassium ions rush out down their concentration gradient, they produce a hyperpolarization of membrane known as inhibitory postsynaptic potential. In short term we call it as IPSP. So inhibitory postsynaptic potential, in other words, they make the inside of the neuron more negative, down to as low as negative 90 millivolt. This makes the neuron less likely to trigger an action potential. So actually the inhibitory synapses they decrease the chances of the postsynaptic neuron to develop an action potential, because kama potential itaanzia negative 70 itashuka negative 90, that means kufikia positive 40 it will be very difficult, and that is the result of the binding of the transmitter substance at the postsynaptic membrane. So transmitter substances themselves are neither inherently excitatory nor inhibitory. So you see kwenye neurotransmitter tutaenda kuona huko mbele, some of the transmitter substances ni kweli they are known as inhibitory, but actually even they are inhibitory but in specific type of synapses. For example, GABA tutaenda kuisoma huko mbele, gamma amino butyric acid, hiyo inajulikana kabisa yenyewe ni inhibitory neurotransmitter, and more especially in the brain. So kwa mazingira mengine ni kwamba neurotransmitter anaweza akawa yeye sio inhibitory, lakini ni ni synapse. So example given here is for acetylcholine. Acetylcholine has an excitatory effect at most neuromuscular junctions and the synapses, but has inhibitory effect on neuromuscular junctions in the cardiac muscles and gut muscles. So you see kwamba acetylcholine yoyo ambaye kwenye neuromuscular junction nyingi na synapse nyingi yeni excitatory, na ndio most mostly use the neurotransmitter in the body, lakini unakuta kwamba kwenye cardiac muscles na kwenye gut muscles acetylcholine yakiwa released ni inhibitory. So these opposing effects are determined by events occurring at the postsynaptic membrane. The molecular properties of the receptor sites determine which ion enter the postsynaptic cell, which in turn determines the nature of the change in postsynaptic potential as described above. So that's all about the inhibitory synapse. And now let's look a little about the neuromuscular junction. Neuromuscular junction usipate shida sana kuielewa, because in some of the exams you will meet with this diagram and then they will tell you to label, or they will tell you what is this, and then maybe explain the mechanism of its function. Neuromuscular junction actually is just like a modified synapse. And here is the junction between the synaptic knob and the muscle. So ni kwamba najua the motor neurons, they go to send the impulses to the effector organs, and more especially here they are glands, and we have glands and muscles. So in the case of muscles, what is formed there is called the neuromuscular junction. So the muscular junction is the special form of synapse found between the motor neuron and the skeletal muscle fiber. Each neural muscle fiber has a specialized region, the motor end plate, where the axon of the motor neuron divides and form fine branches ending in the synaptic knobs. Now the branches lack a myelinated sheath. The neuromuscular junction includes both the motor end plate and the synaptic knob. So we have the diagram here which can help us to explain about the structure of the neuromuscular junction. So the fine branches run in a shallow troughs on the cell surface membrane of the muscle fiber. As you can see in this diagram here, so we have this is the muscle fiber. We have the myofibril, and then we have the cell colema, or the cell surface membrane of the muscle. So this is the cell surface membrane of the muscle. As you can see, it has folds here, and then we have this is the motor neuron with myelin sheath, then axon of the motor neuron. So this is what we call the motor end plate. It is the plate formed by the motor neuron, which when it innervates the muscle. Kwa utakachokiona hapa ni kwamba hapa tutakuwa na presynaptic membrane kama kawaida. Unaona tuna synaptic knob part na synaptic knob. So actually the mechanism of release of neurotransmitters is just like how it happens in the normal synapse. Kwa unaweza kuona tu kwamba this membrane is called sarcolemma and has many deep folds in the trough as shown in figure here. On stimulation, the synaptic knob releases acetylcholine by the same mechanism as previously described. Changes in the structure of the receptor sites on the folds of the sarcolemma, that is the postsynaptic membrane, increases the permeability of the sarcolemma to sodium and potassium ions, and a local depolarization known as end plate potential EPP is produced. So the potential produced here actually is not like how we discussed that it is excitatory postsynaptic potential, or it's not an action potential as such, but it is called an end plate potential. So this is sufficient to lead to an action potential passing along the sarcolemma and down into the muscle fiber through the transverse tubules, that is T system. This action potential results in the muscle contraction as described in the most skeletal muscle. There is one neuromuscular junction per fiber. So other mechanisms of muscle contraction, how these fiber they interact together, and how the mechanism of muscle contraction takes place, actually it's not for your level. So for your level, what you need to know is about the structure of the neuromuscular junction, because some of the teachers, they can ask you about the rules of the synapse, and they want to explain you one among the rules of synapses in neuromuscular junction. And one of other teachers will bring to you this diagram in the exam, and then they will tell you to label some of the parts, or they will ask you how does it work. But actually the mechanism of muscle contraction is not for your level. If you reach in the university, when you be studying about the excitable tissues, then we discuss about how the muscle they contract. So let us end up here in this session, and in the next session we'll be discussing about the functions of the synapse. Functions of the synapse. What we are discussing, they are the rules of the synapse. Now we will go to discuss about the functions of the synapse. Why the synapse is important. Why the synapse is important. So if you are in need of notes, you may just visit my Telegram group, and you may contact me with the Telegram or WhatsApp, or you may use the numbers which are present in the description below to get my notes. But otherwise, I wish you nice studies. Don't forget to subscribe to the channel and share the link for your fellow students. Thank you.